Welcome to the Science Slam, Ocular Indications. My name is Dr. Amit Rakhit. I'm the Chief Medical Officer at BlueRock Therapeutics. And for those of you who don't know BlueRock, we have a company presentation tomorrow, but we're a cell therapy company, a wholly owned independent subsidiary of Bayer organization, and our focus is on neurology, ophthalmology, cardiology and immunology with our key clinical programs, one in Parkinson's for our lead program, and one in inherited retinal diseases in ophthalmology. So with the emerging therapies and innovation that we have in ophthalmology, it's been remarkable to see the gene therapies, stem cell therapies, RNA-based treatments coming forward. And so today we have a panel session. How this will work, we'll have three excellent presenters. Rodolphe Clerval, who's the CEO of Coave Therapeutics, followed by Laura Koivusalo, who's from StemSight, and then Andrew Ramelmeier, who's the CTO at Adverum. Each of them will provide a 10-minute presentation about their company and what they're working on, and then we'll do a 30-minute panel right afterwards, where we'll have a discussion on some of the key topics within this space. So with that, I will introduce Rodolphe to kick us off.
Thank you. Hello, good afternoon, everyone. Thank you, Amit, for the introduction. So I'm Rodolphe Clerval, the CEO of Coave Therapeutics. We are a genetic medicine company with a primary focus on ophthalmology and CNS space. So the company is built on a platform, a new platform for delivery of AAV vectors, technology that have been initially developed in the University of Nantes in the west of France. The company is based in Paris, France. It's a new technology orthogonal to the existing other technology where we basically conjugate ligands on the surface of AAV to improve their efficacy and safety with several NHP data showing superiority of those capsids. We are building a pipeline using this technology with programs in ophthalmology and CNS space with the potential to extend to other therapeutic areas. And we are collaborating with partners that want to use our technology for target indications we will not develop internally. One of these partnerships, there's 4 ongoing, have been disclosed with REGENXBIO. We presented data last year at JSPC. The company is a Series A company. We announced a Series A in January this year for 32 million euro, which was co-led by Novo and BPI. So the platform called Alligator for AAV ligand conjugates is basically a technology inspired from the conjugations of antibody, siRNA, ASO. What we do is we design targeting ligands with a unique linker chemistry to bind covalently those ligands to the surface of AAV, and we had demonstrated recently that technology works on LNPs as well. It's a very versatile technology. Basically, we can conjugate small molecule peptides, cyclic peptides, or larger molecule like aptamer antibodies. And we decorate those AAVs with those ligands, and we're improving by this way the tissue targeting, the biodistributions, and the safety of the vectors. It's a unique technology in the sense that we can play and directly work on existing AAV serotype, first, second generation AAV. We do not change the AAV per se. We can directly conjugate at the end of the manufacturing. It's a one-step chemistry without catalyst. That's why it could be applied directly to AAV. So what we do, as opposed to other companies, we don't screen the capsid first. What we screen is the ligand first. So we design a ligand on a specific receptors or specific mechanism, and then when we have selected the ligands, we conjugate of different serotypes. So bottom line, we end up with a very tiny library compared to other companies where we can screen a few dozens of those conjugated AAV. We can, by this way, be much faster in discovering capsid, and we design those capsid for actually specifically a human target, which is the big difference compared to a competitor in the field where all those companies named here using more or less the same approach, directed evolution, generating billions of capsids that they have to screen on multiple animal studies until they find the right variant without certainty that it will work on human because those capsids have been selected on animal biology. So we have developed conjugated AAV vectors for different applications. Again, primary focus on the brain and the eye. We have capsid conjugated with sugar-based ligands. Those sugar-based ligands actually work by interfering with the natural mechanism of the capsid with certain receptors in the extracellular matrix. So the implication is those capsids spread much better. But there's a secondary mechanism where those capsids, when they enter cells, are less directed to proteasome degradation. So we have capsids that are better in spreading and better in actually transducing and expressing the payload intracellularly. We have developed those capsids for different routes. For the brain, intra-CSF route is one of our lead programs. And in the eye, we have tested different route of administrations with a primary focus on suprachoroidal route, and I will come back to this. We are also developing capsid for IV administrations to target either the brain or the muscle, where here, instead of using sugar, we conjugate cyclic peptides. So those peptides are specifically designed to address certain receptors like transferrin receptor 1 or integrins, and they enable to retarget an AAV specifically to this tissue. Talking about ophthalmology and our lead capsid in suprachoroidal space, why suprachoroidals? It's a very interesting actually route of administrations that could be performed in office. There's actually one approved product currently in the US using this route of administration. It can be performed the same way than IVT. It requires a little bit of training of the physicians, but the physician can do that. It's enabled to target the posterior segment of the retina, which is not the case when you inject a capsid IVT. Most of the capsid actually transduce the anterior segment of the eye. And the consequence of these transductions of the anterior segment, it's increased inflammation because the tissue transduced are very sensible to inflammations. And with the suprachoroidal route, we think that we can have a much better safety profile, and then we can increase the dose if we have the right capsid. There's actually no capsid that have been developed for suprachoroidal route of administration. The traditional AVs are used. Actually, there's a clinical stage program using AV8 for this route, and our strategy was to develop a better capsid. There's actually two programs being in clinic, one from Genetics Bio, one from Korea, using AV8 and AV2, which has demonstrated that by this route they can transduce RPE, but barely not the retina. Our strategy is to develop a capsid that will be able to cross the RPE, but also the retina, and being able to be used for either transducing cells for a biofactory approach, so for a secreted transgene, or for intracellular transgene. So we have demonstrated in the first NHP studies that compared to AV3B, which is one of the best-in-class vectors for suprachoroidal, we increase the transductions efficiency 11 times compared to it in the retina and 21 times in the RPE choroid. Very interestingly, what we have seen is when we inject this capsid on the periphery, we have a nice spreading of the capsid to the macula. There's a second NHP studies that have been conducted recently that is providing even better results than that in terms of transduction of the retina, confirming the very nice spreading of the capsid to the macula. This is in more detail. This, again, small library that have been injected. It is only 5 conjugated vectors that have been tested, a conjugated AV2 and various conjugated AV3B with different flavor of sugars. And all of them demonstrated superiority to 3B, with the AV2 clearly demonstrated being the best capsid to transduce the retina. This is the detail of what we have seen in terms of spreading. You can see that in all the different quadrants of the retina, our capsid has been the best one to transduce those different areas, including the macula, which is a key area in many diseases such AMD. The other very important aspect is we demonstrated on human serum that the conjugation of those ligands, and you have to visualize when we conjugate, we can conjugate up to 500 ligands at the surface. We prevent the actually anti-AAV antibody to bind to the capsid. So this is exactly the same, each number is in the patients, the same serum, which are seropositive to AV9. When we conjugate AV9, we revert the seropositivity of most of the patients are not sensitive to those antibodies. So our strategy is to move ahead with these vectors. We have defined a target vector profile. With a supracaudal capsid, we think this capsid could be the capsid used for developing therapeutic programs with a focus on targets that have been already prevalidated. And obviously, wet AMD anti-VEGF is one of the targets we would like to pursue because there's clearly demonstrations commercially of those target efficacy. There's 3 ongoing programs, gene therapy programs that are also addressing this disease. We think that combining our supracaudal capsid with the anti-VEGF technology will be able to readdress the main issue with AMD, which is the high burden and the frequency of administrations. We think that the ongoing gene therapy will make it to the market, certainly, but at a level of target vector or target product profile, which might not enable them to be widely used because only 50% of patients, what we see today, are off injection free after a year. We think that with suprachoroidal administrations and more efficacious product, we can achieve a better level of efficacy. Thank you for your time. And asking Laura to join.
Thanks. Okay. So hi everyone, I'm Laura Koivusalo from StemSight. And StemSight is a Finnish biotech in the preclinical stage. We're based in Tampere, Finland. And actually our technology has been developed in the local university where there is 20 years of clinical stem cell research right there at the institute. Past 15 of those have been solely focusing on eye applications. And our mission at StemSight is to develop life-changing regenerative medicine products, and we're only focusing on targets where we can actually make solutions that work in the real world. So we're very focused on that. And our core technologies build on our know-how on iPSCs and biomaterials. Our current pipeline is focused on the front of the eye, so corneal applications, where we have 2, for limbal stem cell deficiency and corneal endothelial dystrophy. But since we've been mostly focusing on our lead asset, I'll be talking mostly about that today. So we're currently in vivo proof of concept, which we aim to complete by the end of this year, moving on to GLP tox next year, and aiming to go first in human in 27. So talking about limbal stem cell deficiency, so limbal stem cells are the cells that normally produce a healthy, clear corneal epithelium. And once those cells have been lost, for example, due to burns or chemical injuries, different kinds of diseases, then that results in overgrowth of the surrounding conjunctiva and blood vessels to cover the cornea. And this is a very severe and chronic condition that often affects patients for the rest of their lives. It's also a rare disease indication. And based on the epidemiology data that we have, the number of severe limbal stem cell deficiency cases is 36,000 eyes to treat in Europe and the US combined. And because, even though it's a rare disease, there is actually a high value of cure for this condition. Since the average age of the treated patients is only 34 years, you can imagine the impacts for patients themselves as well as the society. So actually the value of costs is actually quite high, almost $250,000 in EU and double that in the United States. But looking closer at the biology of limbal stem cells in the niche where they reside, so in between the transparent cornea and the opaque conjunctiva. So actually limbal stem cells are located at the bottom of these limbal crypts, where they produce constantly new progeny cells, which then mature and travel all the way across the cornea and to create a layered corneal epithelium. And actually, the challenge with this biology is that we actually lack the specific knowledge about unique targets that would identify this population of cells, these limbal stem cells. There's a pool of commonly used markers such as delta NP63 alpha, ABCG2, and others that have been identified from adult corneal biopsies. However, it's very difficult to identify between the true limbal stem cells and their progeny, which are the transient amplifying cells that they produce. But actually, autologous therapy in limbal stem cell deficiency has been at the forefront of cell therapies overall in the world. Ocular was actually the first cell therapy product to be approved in the market in Europe 10 years ago already. So in this autologous situation, of course, the patient still has to have a healthy eye, and that healthy eye is biopsied. That biopsy is transferred to a facility where the limbal stem cells are extracted, further cultured, and transferred onto a biomaterial carrier for delivery. Then that product is shipped back to the operating hospital and into the patients. And actually, in the original clinical data, the inventors of Ocular noticed that there is a clear correlation between this marker P63 alpha and positive outcomes for their patients. And they said that at least 3% of P63 alpha positive cells in their product resulted in positive outcomes. So that's sort of the history of this particular cell marker. And nowadays, we also have some long-term effectiveness data from 3 years for this therapy. But of course, it still leaves more than half of the patients untreated and has the bespoke manufacturing issues. So we sought to solve this through our iPSC work. And there, of course, looking at two different markers that are routinely associated with limbal stem cells. So first, we were aiming to actually go for a high P63 alpha positive cell population. But what we actually noticed during the differentiation from iPSCs was there actually was an early arising population that was transiently expressing high ABCG2 positive cells. And actually, you can notice from the histogram that they're actually double positive. So they express both P63 and ABCG2. And actually, when we culture these cells further, then they even though they express highly the P63 alpha, they actually are very low in ABCG2. So actually what we then did was find culture conditions that can expand and enrich this ABCG2 positive cell population. And we managed to do that for over 10 passages, just maintaining that particular transient population. So that's at the basis of what, when we started out as a company, we took that academic process and transferred it into StemSight. And what we really did was really drill down on the manufacturing. So we start off with one single HLA gene-edited iPSC line, which we then start to differentiate and then have the enrichment and expansion phase. This whole process takes about 35 days, and we actually end up nowadays with over 99% pure population of these ABCG2 positive cells, compared to the 60 which we started off with. We're currently producing about 100 patient doses per batch, or the scale is at that level, but we also have plans to scale it up further. For these cells are of course frozen, and when a patient would need the product, each drug product would be made per patient by thawing a vial of the cells onto the carrier material, further cultured for 6 days and shipped to the operating hospital. So I just wanted to bring some highlights from our process development showing the cell yields that we've actually managed to achieve, the increase in cell yields as well as the high purity of our cells. A very efficient process that we have actually has resulted in over 80% reduction in cost of goods for our process development. So we're really trying to address this issue from that bottom-up approach as well. And of course, when we want to know what our cells are, so we have done single-cell RNA sequencing to assess both the identity of the cells as well as the repeatability of our process. So actually, when we did a single-cell RNA sequencing from 3 different differentiation runs, we actually noticed that there was a very good repeatability. So I'll show you how these cells during the differentiation cluster together. So we start off with the iPSCs in the bottom bottom right corner there. And then when we start to move to corneal induction, they already start to cluster differently. And then as we move forward with the differentiation towards limbal stem cells and further enrichment passaging steps, you can see how the cell clustering really changes and shifts. And this was very consistent over the 3 different differentiation runs that we did. So, of course, with the identity of these cells being not globally consensus on what the LSCs look like, so we have to look at functionality. So what the function of limbal stem cells is really replacing the lost limbal stem cells in the cornea of the patients and regenerating the corneal epithelium. So when we look at the replacement potential of these cells, really repopulating the limbal niche, we look at colony forming efficiency. And for regenerating the corneal epithelium, we look at maturation, such as stratification, evolution of these mature corneal cytokeratins like cytokeratin 12, as well as the epithelial morphology that we reach when we actually do an airlift culture condition, mimicking the surface of the cornea. Of course, we've done also in vivo work, and we're currently in preclinical in vivo proof of concept stage. So here, this was a study we conducted with University of Pittsburgh. So in this scenario, we actually, in the same operation, removed the limbal stem cells from the mice and implanted our product. And then, of course, we monitored for 6 days how those cells reacted and how the surface of the cornea looks like. And we could already see at 6 days that actually our cells were able to create this layered human cell-derived corneal epithelium that was starting to show a good marker expression of the P63 alpha in the bottom layer of this corneal epithelium. And this kind of in vivo maturation is what we'd be looking for. And now we actually worked in-house to develop a more human mimicking disease model with rats. And now we're transferring this to a large-eyed animal model together with a CRO to show the same histological evidence in an anatomically relevant model. So thank you. Good. So welcome, Andy.
I'm Andy Ramelmeier, Chief Technology Officer at Adverum. I'm really happy to be here. I'm going to mix it up a little bit because I'm a CMC expert, and I'm going to talk to you a little bit about CMC and why it's important. And then I'd like to start off by highlighting a little bit about the eye and talking about our investigational gene therapy product, Ixo-vec. The eye is ideal for gene therapy. It's small, it's isolated, requires low doses of gene vectors that would last a lifetime, theoretically. Luxturna, one of the first gene therapies approved, is a treatment of the retina. A lot of activity in the clinical space, in the ocular diseases, particularly in wet AMD, that's age-related macular degeneration. It's the leading cause of vision loss in people over 65. It's a large indication and growing. The current AMD treatments the global markets are huge, and it's growing to $13 billion in the next 10 years. Now, Ixo-vec, which we are developing for wet AMD, is a novel AAV. It's created by directed evolution. The secret sauce is a peptide insertion, S7M8, that allows the AAV to cross into the retina, where it's transduced into aflibercept, which is a anti-VEGF binding protein that's currently used in the existing therapies. Ixo-vec has been validated in 3 clinical programs, and we're just starting phase 3. One thing I should point out is IVT is a shot directly intravitreal. It's a shot directly into the eye, so you can imagine how uncomfortable and how burdensome that is to patients. The current standard of care requires these shots quite frequently. Now, there have been second-generation therapies that have been developed that reduce that burden from maybe 8 to 10 shots a year to maybe 4 shots. Ixivec, on the other hand, provides a true paradigm shift, limiting potentially patients to 1 shot, hopefully for the majority of our patients. This shows the aflibercept levels in the aqueous humor in the eye for the 4 doses that we studied in our 2 clinical trials. I have 2 messages from this presentation. First, or this particular slide, is that first, that Ixivec enables the eye to make its own aflibercept, which you can see for a long period of time, going on 5 years and counting. The other point I want to make is from our LUNAR trial that the levels that we're seeing in the lowest dose, 6E10, are comparable to what we saw at the higher dose. And based on efficacy and the reduction in treatment burden and our very strong safety profile, why we selected the 6E10 dose for our phase 3 and, if successful, our commercial launch. One of the advantages of the lower dose is now we have a 10-fold safety factor over the highest dose studied, and it greatly improves cost of goods and our commercial supply runway. And I'm going to use this as my segue to talk CMC. I have 4 points I'd like to make. It's very important to have a stable and efficient production system. I will explain why we chose SF9 over HEK293. Because this is such a large patient population, we're talking millions of patients, we'll need the ability to make hundreds of thousands of doses annually at a reasonable manufacturing cost. And we've invested significantly in CMC to make that happen. I will describe at the end, our robust CMC strategy for commercial readiness. There are 3 major platforms that we use for AAV production. I'm going to focus on the 2 most prevalent. That's HEK293 and SF9 insect cell baculovirus system. I'm not going to talk about producer cell lines. I do believe they're the wave of the future, but they're not ready for prime time. There are a number of attributes from a CMC perspective that you need to evaluate to decide which system to use. I've shown these on this slide. It's very busy, but in the leftmost column, are those attributes. I'm going to step through them one by one, focusing on just 293 and SF9. Probably one of the most important aspects of attribute from a CMC is productivity. And that's measured in titer and scalability. And from many AAV serotypes, SF9 has been shown to be more productive than HEK293, generally 10-fold more productive. This slide shows that comparison. This is from data from the literature and also from my team that I worked with at Sangamo. This is experience gotten there. Now there are exceptions like AAV9 where you might see 293 to be more productive. I don't think AAV9 is used for ocular therapies, but for the ones that we've used and I'm aware of, we're seeing SF9 as the best system. From a manufacturing perspective, cost is a very important aspect. HEK293, because of the triple transfection, many plasmids, and also the lower productivity means a lot more lots, and that you're going to need a lot more cost to make the same amount of AAV. It's very important attribute from a product quality perspective to have high full capsid. We see for HEK293, generally it's low, whereas SF9, it's higher. I've seen it as high as 70%, which means greatly streamlining purification, and also, which in the end has higher yield in the end. HEK293 is a human cell line which can propagate human viruses. This is obviously a significant problem. It's less of a problem for SF9 because it's an insect cell line. I think we just, oh, there we go. There are disadvantages for SF9, and those are shown here. Speed to clinic, because you need to make the viral banks, takes maybe 6 more months, adds to your timeline. And then potency, because it can produce, SF9 can produce a AAV with a much lower VP1 ratio, key protein in the capsid, that then generates a low potency vector. This is not a problem for Ixobec. One, we're already in phase 3, and we've already made our banks. And potency, as I mentioned, we made a fair amount of investment in CMC, and we're fortunate that we're able to have a high VP1 ratio in SF9 and a high potency. You put it all together, you can see why we selected SF9 for Ixobec. Okay, this is, I want to wrap up by talking about dose and cost of goods. And this has been a huge challenge for gene therapy, particularly the systemic delivery. Now, you see very high doses, which I think that comes out to be about 1E15 per dose at a very high cost. Now, Luxturna is an exception, but that's for the eye, and I'll actually talk about that in a second. But because for these systemic systems, cost of goods have been hundreds of thousands of dollars. Now, I've seen this developments over the last 5 or 10 years. It's starting to bring that down to under 100,000. Now, for gene therapy of the eye, it's a totally different situation. With the lower dose that I just highlighted, and then combine that with our efficient production system, we have very low cost of goods, which will allow us as product to be very cost effective and potentially cost saving. Okay, I think I might need someone to do, okay, here we go. This is just going to wrap up with my final slide on our CMC strategy. And this basically shows how CMC is positioned well to support Ixobec in our pivotal program and commercial launch. I already talked about our efficient process that we developed in-house. We manufacture that currently at a global CMO at manufacturing scale. We have commercial analytics, and we're testing in-house. Our Ixobec phase 3 material has already been manufactured, and we've been working very closely with our regulators as enabled by our RMAT staff with the FDA and PRIME designation with the EU. And this concludes my presentation. I'm very excited, very proud to be involved with bringing Ixobec to patients. Thank you. Are we staying? Do you want me to go on? Yeah. Okay. I'm going to grab my water because I'm starting to.
Great. So as Andy's coming up, well, thanks for those very interesting presentations, and we'll spend 30 minutes just talking about some of the, I think, challenges that we see in this space. I think each of you, maybe we'll start off, we've seen so many new therapies come and a lot of interest into the ophthalmology space with different modalities, different approaches, gene therapy, cell therapy, RNA therapies. Maybe start at the individual level with your companies and then take it to a macro level. I would say, from an individual perspective, what are some of the challenges, whether it's scientific, regulatory, talent that you're facing as you're thinking about bringing your therapies from the lab, the research, into the clinic and to patients on an individual perspective. And then on the macro perspective, these different modalities, and many of us here are working in this space also, how do you see these different modalities either complementing each other or competing with each other in the broader ophthalmology patient treatment space? Laura, you're right next to me. I'm going to start with you.
Perfect. Yeah, of course, for us, I think the main challenges that we've been struggling with at the company has been the translation of within the preclinical setting, since that's where we are. And I think for us, a lot of it has been focusing on the animal models, because we've had to develop our own animal models. So that has been challenging. What we've found as sort of the key components of how to make it work is really to nurture the talent in-house, to actually bring knowledge in and really keep that knowledge. So I think that sort of persuades like all through the company. So we actually have now adopted a view of actually, even within the regulatory perspective, so just taking talent in and learning from experienced talent and keeping that knowledge. I think that's sort of where we're at. I think for, like you mentioned, for the broader perspective of how we can complement each other within the cell and gene, or both cell therapy and gene therapy. So I think there is a lot of complementarity there. For us specifically, of course, we're focusing on the cornea, where there's not just such a clear gene therapy advantage, because we have a broad range of causes for our disease. So of course, replacing lost cells just makes a ton of sense. But I do think that also for the retina, we do have some scientific background in that as well, so I hope to speak a little bit to that. So I think for how I see it from my perspective is that gene therapy is like an early fix, that when there's still something to be fixed, and you can sort of go in and try to fix it with the tools that you each have like your own. But then again, if you've already lost the cells that you sort of need, then perhaps these kind of later stage developments or later stage rescue missions would be cells.
Rodolphe, how's that different in the more of the gene therapy space?
Well, just to follow up on the last point, I mean, I do agree that most of the gene therapy program we see today in ophthalmology really address not late stage form of the diseases. We either replace a gene to prevent the loss of photoreceptors, or we do supplement with an anti-VEGF or other molecules to prevent the destructions of the retina. There's few programs that are looking to using optogenetics to revive or to switch some remaining cells. I think this is probably where cell therapies, from my perspective, make much more sense. Talking about the regulatory development challenges, so the company has an interesting history. The company started with rare disease of in the retina. We have developed a program up to phase 2 that we spun off to a separated company. And for this rare inherited retinal dystrophy, the main challenge is actually how to bring those products to the patients with an accelerated path in terms of regulatory. We have phase 2 data on 17 patients. It's a slow progressing disease. It's called PD6 retinitis pigmentosa. We know for that disease that if you want to see, and we have seen clear efficacy of the product, we have to wait 5 years, where you really see on an approvable endpoint the acuity that there's a difference between the treated and untreated eye. So it's not just possible to run a phase 3 for that kind of programs. I mean, it's not feasible because there's no capital to do so. I mean, we have seen that for many. So today, I mean, if you look at in ophthalmology, there's probably this is the area where in gene therapy you have on one side approved product for rare disease, many products that have compelling clinical data to be approved, and in few years, product being developed for more prevalent diseases like AMD, and this is fantastic, but for inherited retinal dystrophy, really there's a challenge in terms of how the FDA and the European agency could help companies to get those products approved on the basis of the phase 2 data, basically. For more prevalent diseases like AMD, I think the main challenge for a company like us will come as a second generation after Adverum for DMT, where those companies will probably have approved the product. And there's a lot of competition. So we are in this situation that other companies face in hemophilia, where when you try to beat the standard of care, the challenge is how this standard of care will change over the years, and the bar will raise, and how you adapt your development to be sure you will be able to at least to be in a non-inferiority demonstration. So this is the main challenge I think we'll face for that kind of indication.
That's great. Anything to add?
Yeah, so I'd say some of the biggest challenges is the fact that we're going to be treating a large market, right? A lot of the systems have been set up for the systemic gene therapies for orphan drugs. We need to do things differently. But at the same time, it is, because it's in the eye, there's a real opportunity, to drive this to a different place than it has been currently. Like gene therapy is kind of an oddity. This could be an opportunity where it's going to meet, there'll be millions of people that could benefit from this, right? So we need to set up those sort of systems. We also need to educate, hey, this is different. It's not the same old. But I have to admit, regulators love it. We've had very good interactions with them. They see the promise, and they really want this to work. Our patients love it, right? I'd say 95% of our patients want this therapy because it's so burdensome, the existing therapy. So we're really excited about that. But there is some challenges, particularly to commercialize this, that we have to overcome.
Yep. So I want to go back to one of your slides I saw, Andy, on manufacturing. So, and I know a lot of people interested in this space around, I can develop a lot of things. I'm more on the development side and clinical side. But I found in my experience, and my little experience I have like in cell therapy, because more of my experience is in small molecule, but in cell therapy, it is critical, the manufacturing CMC. That is probably the bulk of the work, the research CMC manufacturing. And once you have that right, I can go down the clinical path through phase 1 through 4, but it's really critical that you have the right cell line, you have the right gene vectors, you have all of that worked out. And so, and Rodolphe, you mentioned that you've kind of in this rare disease space, orphan disease, sometimes that's a little bit easier with scale and just getting that kind of initial pieces right. But there is this larger patient population, and you showed the market potential, and I think yours did also, about billions of dollars, millions of patients out there. So what are your approaches? How can you get suitable scale for that patient population that we all want to address, in the future? But what are you putting into place now? How are you addressing manufacturing challenges? What are you seeing there? What are some of those pieces to get that right, to get to the right scale, but balance with the COGS and just ability to do so, right? So you mentioned it, and all of it came through in all of your presentations a bit, but just maybe we can touch a bit about that, because I think that's a really critical piece of our space.
Well, I'll be brief because I kind of went through our spiel and why we pretty much, I think there's a few more things we got to do, but we pretty much got CMC in a good place. And for ocular gene therapy, because of the dose, that's a great game changer. You still need an efficient system, particularly if you want to get the cost reasonably, but you also need to have something that's robust, right? And that's probably one of the things that they really should be focusing on, finding a good system, a good cell line. Right now, there was a time when people were using adherent. That should be the thing of the past. And that's a challenge. Suspension cell culture is where things should be. Now, cell therapy, I'll talk about it, only that I spent a lot of time at Sangamo. It's obviously a challenge. I worked on a regulator T cell. Now, this isn't for the eye, but it's a very complex process. And doing it autologously, it's a challenge. And you kind of wonder at some point, does that really make sense, right? We need to try to simplify things, right? And that includes manufacturing. And that's going to be the challenge in any of these situations, is trying to simplify. Automation is good, and I agree with Daniela's point. You can't automate a bad process. It does start with a good process scientist to make sure you've got the right process.
I think if I'll jump on the queue for cell therapies, and I think for us, when we started, majority of the work that we've been doing for the past 4 years has been in CMC, right? To get the process where it needs to be. And even though we're starting off with a rare disease where the number of cells is not that huge, but we are looking at bigger indications in the future. And for example, there we start off with the premise that, okay, we just cannot do cell sorting, for example. So we just cannot. We have to find other ways. And when you sort of take those as sort of a base level assumptions on your process, then you start to think about, like, how do I drive the efficiency of my cell yields, and how do I drive the scale? So I think it's sort of, you have to build that in from the very beginning.
Just to echo the comment from Andrew, I mean, it's true that for gene therapy in ophthalmology, the doses are so low compared to other tissue that, I mean, most of the clinical stage programs using traditional AAV or first generation AAV, I wouldn't say they don't face any problem, but we know that the yield we achieve already enable a cost of goods that will be on the ballpark of what it needs to be. Now, when you come with a new technology that we do, where we have, we're using AAVs, and on top of that, we conjugate small molecules on the top of it, we have to be sure that this technology will enable to achieve this yield and this cost of goods. So we, very early, 3 years ago, when we started this platform, we have hired a team of developers, process development teams. We internalized that. I think it was very important that we have internally the capability to demonstrate we can scale up the process. And we have developed the process demonstrating that actually the way we do the conjugations without changing the traditional AAV manufacturing, just embedding the conjugation between two existing steps without any additional purifications, enable to achieve the same yield. So that was already a demonstration, and we are collaborating with big pharma and biotech, but before we generate those data in manufacturing, they were like, whoa, you are adding complexity on an already complex manufacturing process. So we had to demonstrate them. We have scaled up the process in GMP conditions. We released our first batch, and they were really, we were all very happy about what we demonstrated. The second things we did very rapidly, we went for pre-IND meeting with the FDA and the European agency to discuss the CMC package, and especially the new QC we have to develop to characterize our products. So we were not starting from zero because there were a ADC that paved the way about what kind of a QC you have to establish to demonstrate how many ligands you put at the surface and how you really measure that on an accurate manner. So we did all this work, but we still are paying attention because now we have to scale up more the process. But I think taking very early in the development of a new delivery technology, the CMC aspect is critical. Otherwise, it will never end up with a commercial product.
Yeah, absolutely. I'm curious because both of you also, like, the rare disease indication comes up quite a bit. How did you actually go about thinking about your indications? And you could have gone into a larger indication rather than rare, right, for various reasons. But how did that thought process kind of, as advice to maybe a new startup company in the cell or gene therapy space that's looking and has some promising new therapies, how should people be thinking about which indication to go after?
Well, I think for us, when we were working with iPSCs and developing a differentiation process. So of course you have to know what type of cell you're aiming for. And for us, basically the need came from the ophthalmology community locally, and they actually said that there is an unmet need. They have patients they can't treat. Even though Ocular was sort of getting to market, it never reached Finland because of the long logistics chain. So for example, that's sort of what drove us. And then when we started to crunch the numbers, we actually see that it was a business case, and it was sort of easy to build around it. So that was sort of an easy access point for us to showcase the technology that works.
And in terms of patient engagement, and how early would you recommend? You see the gamut. I've seen people who start very, very early, very late, in the middle, but how has that experience been for you with the ophtho committee and the patient and community perspective?
So for us, we haven't done active outreach to patient communities. There's not so many patient communities within this disease. However, we have had patients reach out to us particularly, and we of course are so sort of impacted by that. It's what keeps us motivated, basically.
I will, in my last company, Sangamo, once they announced an IND, they started bringing patients in. So I've been engaging with your key patient advocates and starting those relationships. You got to start from the very beginning, right? So I will say that.
Yeah.
So from again, our previous experience, going to rare disease in the eyes, I would recommend a company to be sure that there's a path, there's a possible rapid readout in clinical trials, there's a path for accelerated approval. And let's cross finger that the pediatric breakthrough designation of PRV will be revived at some point, because this is critical. Without this kind of incentive, I would not recommend today. I'm really speaking about financing a company developing a product to go after a ultra rare indication as we did, unless those elements are on the table.
Also, Rob, can you comment on your kind of FDA experience, as well as if there's any differences between some of the approaches to gene therapy as you're looking at this in different regulatory markets, like Europe or Asia, if you've gone there or have a sense of that? You mentioned the FDA. I know there's changes happening at the FDA, so at least whenever you went, how was that experience? And I think what was your takeaway?
Well, our experience, I guess, is pretty similar to others. We had, in the context of pre-IND, to face the FDA and the European agency on that. And actually, we have seen the FDA being much more pragmatic, especially with respect to new technology like ours, and much more open to support us. Obviously, that was very clear. Doesn't mean that the European agency is not positive. They are hard, but FDA, I think they are more exposed to this kind of new companies coming with new delivery technology and asking about what would be the package they want to see.
Eddie?
I can't comment specifically, but I will tell you that there are still differences between EMA and FDA, and I know they want to bring them together. And when Emer made the point that they have all these workshops together with the other agencies, and I'm like wondering, do they have like a helpline where a company can say, hey, we have this issue, can you help us resolve it, right? I just wonder if, because to me that would seem like it'd be better to be talking to us rather than talking to each other. But that said, there's still work that needs to be done.
You should have caught Emer before she left.
I should have asked that question.
Anything else to add, Laura?
No, I think so. We're just approaching the FDA now this year, so.
Okay, great. Maybe just about people, right? I think that's also, given this is fairly new areas, not so new anymore, but still, cell and gene therapy, finding talent, finding the right people who are a good fit for your company, but also have the skills and knowledge that you need to be able to be successful and grow. How are you approaching kind of people, talent? You mentioned that you bring people in-house and train and get that in-house experience, which is great. But sometimes you still need somebody that and you're looking out in the wide world. How are you approaching that? What are you finding? what are some of the takeaways that you've seen from that?
It's hard, right? I mean, I will say when I tell all of the people in my team and everyone in the company that if you have a position to fill, that's your #1 job is that getting that done. Because if that position isn't filled, either you're doing the work of 2 or something's not happening. So this needs to be your priority. We have a really strong talent team. That's been a benefit for Adverum, and I have to admit we find some really good people, and a lot of times we're going to have to go remote, right? That's, obviously COVID did kind of allow that. Before it was not, was never, I don't think anyone would consider it. But now we have, I'd say 20% of our staff is remote, right? So to get the talent we need.
Do you find that be a challenge sometimes? Because not everybody can be remote, right? Labs people usually have to be there, but
we can't.
A mixture.
Lab staff can't. Manufacturing staff can't. But actually, my head of manufacturing, or our head of manufacturing, is in Philadelphia. And in the end, he's very effective on, and he does travel, right? So.
It's interesting. We've had some, not challenges, but just creative approaches to people who can't be remote, because there are people who can, and if they are choosing to be remote, you want to be able to help kind of manage that expectation.
Finding the best people, that is, as a leader, that is that's our #1 job.
I think if I can comment on this, being based in the far reaches of Europe in Finland, so it might be a challenge. However, we've actually haven't had a problem with it. I think for us, of course, the people who actually need to be in the lab staff, there's actually a very good supply of very experienced, very good hands-on talent, because there's not a lot of competition around that talent. There's just academia, and then what? So it's sort of, the biotech industry is small. However, for the more experienced, the seniors, the people who actually know how these are done, those we've had to find outside of Finland, and that's where we have worked remotely, and we have like 2 people in the UK, 1 in Australia, and that works. But they're all management, so.
Well, actually, so from a European company perspective, the things have dramatically changed over the last 2 years. I mean, we have plenty of people being available, and what we've seen recently is plenty of US-based European people having the desire to move back to Europe. So we have, I mean, the thing is, a lot of these European profile in US have been in US for quite a while, 5, 10 years, and with a sudden, crisis, and they lost their jobs, and they have to find options, and we just recruited 3 people from US that are moving back to Europe, and that's great. And it was much more difficult before because, of course, the salary package are not the same, but the cost of life as well is very different. So it's a very great opportunity, to be honest. We are not using headhunter anymore under the timing, and that's, I'm sorry for them, but it's a very good moment for a CEO to recruit.
Absolutely. Well, I see the timer running down, so maybe just one takeaway that you want this audience, having kind of been here representing all aspects of CGT, but maybe one takeaway message that you'd like to leave the audience with. Laura, I'll start with you.
Sure. I think, since we are in an ocular indications panel, I really want to highlight the why it matters and why ophthalmology matters. Even though it's not life-threatening, it's still our most important site, and it is really life-changing for our patients. And as a one thought that I could leave with the audience is actually ophthalmologists are the most keen to adopt new therapies and new technologies. So it's a good target audience.
Great.
Yes, from on the perspective on gene therapy, as I said previously, the ophthalmology is really the area where we are seeing at the same time product in rare disease with clear efficacy. One have been approved, and very soon other will be approved because they are in an accelerated approval process, which is great. And at the same time, it will be very likely the first therapeutic area where we're going to see blockbusters in therapy for large indications such as AMD. And it will happen in the next 5 years. So that's really where the ophthalmology is forefront of demonstrating the commercial viability of gene therapy.
And Andy, one last thought?
For ocular indications in gene therapy, it's going to be a game changer because of the ability to reach more patients. And as I highlighted, we're going to hopefully start seeing meaningful products, blockbuster products that will help drive some of the things that were talked about in the prior session that'll be the catalyst to kind of get people excited about gene therapy again.
Great. Well, thank you, audience. Thank you to our panelists. Big round of applause to Andy, Rodolphe, Laura. Looking forward to great things coming from you guys soon. So thank you so much for joining.